Hello, everyone. Today we're going to finish up protein synthesis. So, we're going to talk about a little bit about transcription. We're going to talk about translation, which is a new thing. And hopefully, we're going to kind of wrap up some of those loose ends you guys might have had after talking about transcription. So, this image right here, you guys have already seen before. We talked about how this is the cell membrane, we got the nucleus, we got some DNA, and then from the DNA, we then do transcription, and this is where we're going to make our mRNA. Now, we have talked about mRNA, how this is the messenger RNA, and this is going to take that message, that gene message, it's going to take it outside of the nucleus and put it into the cytoplasm. Now, when it gets to the cytoplasm, this is where translation is going to take place, and that's a new step we're going to talk about today. Now, for everyone that's had me, we're going to talk about a cookbook today. That's the backside of it. We're going to talk about a cookbook today as we get through this. I'm going to refer to things as recipes and translating recipes. Now, if you don't have me, I will make sure it makes sense to you. I promise. Don't worry. But it is a way that I like to introduce this or talk about transcription and translation that just makes the most sense to me. Maybe it's because I'm a big guy and I like to eat. I don't know. But for some reason, it works. Okay? So if it doesn't work for you, I apologize. That's how we're going to do it. Let's go. So first and foremost, guys, you've already seen this before. You've seen DNA. You've seen how we got to transcription. So all of a sudden, our A's go to U, our T's still go to A, and our C's still go to G. But this part right here, the words amino acids, you might know. It is the monomer of a protein, which is the polymer chain that we've talked about before. But TRP and PHE and GLY and SER, that's a bunch of gibberish. So today we're going to actually talk about what the heck that means. So, first and foremost, the first thing you need to write down, translation is the message from mRNA is translated into an amino acid. Now, how do we get those amino acids? We're actually going to have something called a codon chart, and I'm going to show you guys that a little bit later. But for the people that don't know about this cookbook, let me tell you. So what I told my class is that this cookbook is a DNA sequence, okay? Inside of here, there's a whole bunch of different recipes, which are going to represent our genes. So, for instance, I don't want that one. That one's gross. Let's choose this recipe right now for some cube steak. This recipe is a singular gene. Now, let's say that Miss Knoll really wants this recipe, but I'm kind of a stingy person. I'm not going to give her this whole cookbook. So what I'm going to do, and being kind of a butthole, I'm going to write this recipe for her, and I'm going to give it to her. But there's going to be a twist here. I'm going to take this recipe, I'm going to write it in Russian, and then I'm going to hand it to her. So now she doesn't have the cookbook. She doesn't have my original DNA sequence. She does have the recipe, but she's going to have to translate it. So she has everything. She left my house. She has everything. She doesn't have the original DNA sequence, but she has the recipe. So she has the mRNA. And now she's going to take that messenger RNA, that original recipe that is now in Russian, and we're going to do translation. And that's kind of how we're going to cut up with this. So what we're going to talk about are something called mRNA codons. So there's going to be three bases on mRNA are grouped together from left to right to form one codon. So real school, my bad. Every three pieces of a section of mRNA are going to be considered a codon. So AUG is one codon. AAC is one codon. If you see a T here, anywhere in here when we're talking about codons, you did something wrong. Go back, double check, get rid of the T, make sure that you put a U. Okay, but AUG is one codon, AAC is another codon, and this will happen if we have, say, 30 pieces of mRNA or 30 sections of mRNA. That means we have 10 codons because 30 divided by 3 gives us 10. Okay. Now, we're going to introduce two new types of RNA. We have rRNA and we have tRNA. So we already have messenger RNA, which is the original mRNA. Now we're going to add rRNA and tRNA. So rRNA reads mRNA and matches it with its tRNA. What does that actually mean? So if we look at this image over here, this blue line is our mRNA. It has our codon, codon, codon. You already realize that everything's already switched to U's, no T's. And then we have this big blob that almost looks like a big puffy donut laying around it. That donut is our rRNA. And what this is actually going to do, it's going to match these tRNAs. It's going to pull them inside, and they're going to put their anticodon on top of the mRNA's codon. Now, that's a couple new vocab words, and let's talk about what these actually mean. So this thing right here, which kind of looks like a T if you like kind of squint and look at it like this. Okay, it looks like a T. They have base pairs on the bottom. These base pairs correspond with mRNA's base pairs. Now, mRNAs are called codons. tRNAs are called anticodons. Okay. Now, the tRNA also carries an amino acid on top. So what's going to happen is that this mRNA is going to keep working through this rRNA, and the rRNA is going to grab these tRNAs, and it's going to bond them too. So the UUC corresponds with AAG, so the codon bonds with this anticodon. Now, when this happens, the only reason we're doing this is to make this amino acid chain. So once this tRNA is over with, the tRNA goes away, the amino acid chain stays, and guess what? Now we have our amino acid chain, which is actually going to make our protein through something called protein synthesis. Now, I know that's a lot. We have mRNA, we have rRNA, we have tRNA. We need to make sure this actually all makes sense. So what is going to happen here? We're going to take this recipe, is my original piece. I already transcribed it. I gave it to Miss Knoll. So now she has this recipe and she has to translate it. Now, I'm assuming Miss Knoll does not know Russian. So what she's going to do, she's originally going to translate and she's going to find what her codon from the recipe is equivalent to for her anticodon. So let's say in the recipe book, you're supposed to have potatoes. But she has to figure out what potatoes is in Russian so she can translate those two things. Now, when she figures it out, she then can buy the ingredients, which is the amino acid. And then once you put all the ingredients together, we're actually going to end up building a protein. Okay, or we're going to end up building the recipe or making the recipe. Okay. So moving on, a chain of amino acids is going to be called a polypeptide, and this is going to fold into a specific protein. Now, like we've talked about already, every single protein has its own distinct shape. And it has its own distinct shape because it does its own distinct function. Maybe your teacher said this before. I know I have said this. You wouldn't buy a glove to wear it as a hat. What I mean by that is everything has its own specific function, and its shape dictates its function. A glove wouldn't fit on your head. It's not the right shape. That makes sense. So guess what? You're not going to use it as a hat. Okay? But when you do this, we're going to make that specific protein, and now we have that protein. Or in other words, we now have this completed recipe, and Miss Knoll can eat the food that she made. Okay. Now, how do we actually figure out what amino acid represents what? We're going to do something called transcription, and we're also then going to translate it. And here's this big chart. So let's just do what we already know how to do. We have our DNA sequence. We have AAC, and we have CTT. Let's just transcribe this. So let's make our mRNA sequence, which should give us our UUG. Whoa, my bad. Our UUG and our GAA. Now, what the heck do I do to figure out my amino acid sequence? It's actually kind of like playing Battleship. It's not too difficult. So what we're going to do is we're going to go to a U. We're going to go to U. We're going to find the G. Boom. UUG stands for LEU. Then we're going to go to G, which is down here. We're going to go to A, which is over here. GAA, which stands for GLU. And guess what? Boom. LEU and GLU are both of our amino acids we need to do. We just did transcription, translation, and now our body is able to fold this amino acid chain or this polypeptide chain and actually make a protein. Okay? So we went through the process of making messenger RNA. So using that recipe to make another recipe. We then went into translation. So how we actually can translate a recipe or translate that mRNA with rRNA and tRNA to make a polypeptide chain, which then folds into a protein, which gives us our proteins at the end. This is the whole process of protein synthesis. Now, you guys are going to work with this a little bit on Friday as well, but here's a couple things for you that you need to make sure you understand and make sure that you do properly. First and foremost, when you transcribe, make sure you use U's. You. Okay? So A goes to U. No T's. As you notice right here, there is no T's in this thing. Do not use T's. That's step number one. Step number two, make sure you read what something's asking you to do. If it's asking you to transcribe, that means make your mRNA. If it's asking you to translate, this is telling you to get the amino acids. So then you have to use a codon chart. Now, this might also be shown in a codon wheel in some different images. Same premise, same way to use it. Do your best. If you have questions about this, please reach out. Please ask. If my cookbook example made absolutely zero sense to you, I apologize. You just listened to me talk a whole bunch about some food. Maybe it's lunchtime. Maybe I'm hungry. I don't know. If you guys have any questions or concerns, please reach out to your teachers. We're trying to do our best right now, guys. Please let us know. Anything else, just reach out. I'll talk to you guys later. And let me get my image out the way. And bye.